So we have three individual presentations and one joint presentation. And I'll let Tamaki introduce them.
Lovely. Thank you so much. So yes, so now we are moving on to the 3 R's in your curriculum, research, and the professional practice. So we have exciting presenters from different stage of their research or career stages. So first, we will start with Matt. Matt is a SCM student at the Molecular Microbiology and Immunology at the School of Public Health.
Hello, Tamaki, everyone. Thank you so much for having me. It is an honor to be here. And today I'm going to talk about, living the R3 curriculum in my, both schoolwork and in my thesis research. And I will share my screen now because I have a PowerPoint presentation for you guys. So give me just a minute to see if I can pull that up. There we go. Can you guys
Perfect.
see everything?
Yes.
Okay. Okay, so I am here. As I said, my name is Matt Seibel. I'm a second year MMI SCM candidate, and so I'm going to talk about this stuff today, and I'm really excited to talk about it. First things first, I'm going to talk about what I've currently done, like what the R3 curriculum that I've taken and stuff like that, just to talk about it a little bit. So I first took How Do We Know in term one of my first year, and then I took Causation and Anatomy of Scientific Error in my fourth term, and I'm actually currently the one of the two teaching assistants for Causation this term. So I definitely have a lot of experience and a lot of work going into these things and a lot of these classes. And so I'm like, they really inspired me and they really helped me think about a lot of different things and like how I go about my science and my research in general. And so, like, some of the reasons why I think these classes are really important to me was because the knowledge and skills that I've gained, I apply them in ways that I don't think I would necessarily have done so if I hadn't taken them, or like if I just relied on a traditional classroom or lab basic science setting for my graduate education. And I've also been able to apply these skills towards my professional research interests, which gave me a more well-rounded understanding and a better view of all the topics I'm interested in a critical lens. Like, for example, in causation, my thesis is related to antimicrobial resistance in mycobacteria. So for when I was in causation, I decided to look at, all the different factors and all the different reasons that, antimicrobial resistance, things that cause antimicrobial resistance. And I think that kind of gives me a more greater understanding and a more greater appreciation for my work as a whole, but also like being able to sit down, critically think about a topic and like kind of go forward and work with things. And then I just get a greater appreciation for my own research because I know how important the applications of rigor, reproducibility, and the responsibility of, producing accurate and good science is, It's like I do a lot of rigor, and I'll talk about this a little more. Like, I actually want to talk about it right now. Can you see my mouse? No, you cannot. But so that's why I circled this part. So this is my main research method right now. This is what I'm doing for my work right now. And I circled the bottom corner because I feel like it is really, really important. Because this highlights both rigor and responsibility in science. The current literature usually says that colonies that grow on an antibiotic supplemented inoculum tend to all be resistant. But in my work, we're taking it an extra step further, and we're actually testing like a subset of these colonies and testing their MIC values to see if there's actually an MIC change to be resistant or not. And we're actually finding out that they're not. They're not resistant. A lot of them actually have the same MICs as the wild type strain that is technically considered susceptible to these drugs. So it's kind of challenging and adding new context to something that was originally done, like something that the literature currently does that's different. This is a really, really rigorous process. It takes a really long time to do these. Takes me about 2 to 3 hours to do one set because I take 20 different colonies from my drugs of choice, and but it comes out to a lot more accurate math numbers in terms of resistance and persistence, and I think that's a really important a thing that we should be doing going forward because it is showing this science what we have been doing is actually not necessarily the same thing. and not actually what is correct. And I think that's a really, really important part of like the rigor aspect of the R3s. And then, so like in my work, I was talking about this. Traditionally, when bacteria are inoculated and incubated on media supplemented with antibiotics, we've initially assumed that all colonies are considered resistant mutants. And the phenotype on media is actually resistant, but is it actually though? Is it actually? And so then that's why we decided, what if we looked at the phenotype of this in the context of MIC testing and not just growing on a plate? And so therefore we did this. We found out that actually for a lot of these, not all of them, but for a lot of these, we are finding that they have the same MIC as their sensitive wild type strain counterpart. And so like the additional rigor allows for more accurate readings and actual resistance determination. And this is also something that I had to do a lot of, because before I even started my research, like, yeah, there were MICs in the literature, but I did it myself. I went through, took all the drugs that I was using, did MICs, did double replicates, both biological and technical, to make sure that I was getting accurate, reproducible results before I could continue. Because the backbone of my entire research project is based off of these MIC values that I was getting. And so I wanted to make sure that it was as accurate and as reproducible as possible to me and to my data, so that way it was not showing any issues going forward. And I think having taken the R3 classes, I get a greater appreciation for being able to do this, because honestly, if I hadn't really taken the R3 classes or thought about it, would have been like, why can't I just use the literature? Why can't I just use those values and then go forward with this? It would have been done much faster. But no, I actually, now it's having that first step and having that literature, like I did that stuff. That was my work. I can say I did that. I found these things, and it contributes to my research much better than if I just said, oh, I found these MIC values in the literature and cited them and just used those. I feel like it just gives a stronger backbone to my research as a whole and makes it a lot more valuable. And then, so this is the importance of critical thinking and the reduction of biases in my work. So fun fact, I was actually going to read about my MIC essays, but I was not allowed to read the literature on mutations. There actually are a couple pieces of literature on like how frequently the bacteria I work with mutates in response to certain antibiotics. But the idea behind this was, at least when I talked to my PI about it, because I was like, why? Why are you making me do this? Like, why was I not allowed to do this? And he was like, because I want you to be able to sit down and analyze your results with an open mind. Like, I want you to look at your results and analyze them yourself. You don't want to jump to conclusions or think XYZ just because a paper said so. If I'd only relied on the literature to guide my result expectations, I could have opened myself up to like different types of biases, most specifically anchor bias, which is when your brain like kind of knows something and you're kind of expecting a pattern to occur over and over again. And this is not me saying literature is bad. I actually think the literature is wonderful and it's a really, really good thing to have. But I am not saying the literature is the end all be all of everything. The literature is very important, but you need to be able to also think for yourself and have an open mind going into looking at your results and analyzing them, because that can sometimes lead to novel and potentially serendipitous discoveries, which I know is a thing that we do discuss in one of the R3 classes. And this is not to say, obviously, that the literature is not valid. I think it's really important. But when you look at your results are new data, and you don't want to be like, oh, well, like this paper did this and it got a different result from me. My stuff is wrong. My stuff is trash. Like it's not important or it's not good. You need to still sit down, look at your results, and think critically about them, and be able to report them in a way without saying, oh, well, this didn't work, or why didn't this work, because I was expecting this because the literature says this. Just because the literature says this doesn't always mean it's true, especially if you try something slightly different. If you change the protocols a little bit, you could always get a change in something. And that's also how that's how like new discoveries are sometimes found too, like, and like the science is always changing. You might have new methods, you might have new technology that like gives us a different answer than what we currently have. So like there's just a lot of different things you have to think about taking the R3 classes, working with the R3 classes, and like being able to just kind of think about how important these are in the context of being a scientist is really important, because I feel like if I hadn't taken these classes, or if I hadn't, done as much, like if I went to a graduate program that didn't have these kinds of things like, I wouldn't have been considering or even thinking about these things as much. And I feel like that's a really important topic to hammer home. And I'm really am grateful for it. And like, I'm also really grateful to my PI for kind of like, he's not in MMI, he's a School of Medicine professor, but he seems to kind of echo and resonate a lot of these topics and a lot of these thought processes. So like I've been able to kind of, take what I've been learning, and I'm able to like use them as a backbone to bounce off, or like kind of put what he's putting on me to understand and explain things and how to think better. I obviously I'm coming from the R3 more from a critical thinking lens and more of like a rigor in science lens, but there's a lot of different things about it that are more than just that overall. As the student panel had said earlier, there's a lot of different overall encompassing things. But at least in terms of my research and how I'm currently using it, my focus is on the critical thinking and the rigor aspects of it. And then my research overall, like, I want to make sure that it is accurate and it's, good, because the responsibility, if this works the way that my PI and I think I will, like this could change the way that current treatments are used for Mycobacterium and abscessus infections in the clinic. So it's very important to also be accurate and responsible to give that research out in an accurate manner. Anyways, thank you. And
Thank you
then
so much. Thank you so much, Matt. And we will move on to the next presenter. It's going to be a duo presenter, Sumina and Kristen. Floor is yours.
So I'm Kristen. And I'm Samia. I know you guys heard from Kristen already, but I'm also a PhD student in MMI in the 4th year cohort, also in the Persad lab studying perinatal HIV. So we'll be talking. I know his presentation before us, he kind of went through some of the classes that he's taken. So we also have TA'd many of these classes before, so we thought we'd provide just a little bit of background of what the classes actually provide, but then spend a lot of time at the end for our specific research, how each of the three R's relate to what we do. So we can start. As Kristen said, we completed the R3 certificate program, so rigor, reproducibility, and responsibility in scientific practice. This program provides an introductory graduate level training in epistemology, logic, ethics, as well as quantitative and communication skills. The program specifically addresses the 3R core norms of good scientific practice applied across the health and science disciplines, including fundamental and transdisciplinary approach skills and then you can see the rigor, reproducibility, and responsibility. So we'll first start with our kind of like the introductory course that most students take, which is how do we know the theory, history, and practice of science? Yeah, so this course is designed to first examine the foundation of science using interdisciplinary approaches, emphasizing critical thinking and storytelling, explore revolutionary discoveries in science and public health, discuss the principles of good science practice. So again, looking at the 3 R's. And then lastly, considering the relationship between theory, practice, and also serendipity in the scientific discovery. And so the session topics that this class offers is what is science, so looking into evidence versus belief, revolution or evolution, so what marks a paradigm shift in science, what is truly revolutionary in science, and then going into like the scientific method, which I think all scientists should be well aware of. How is it developed, and is there only one for all or multiple scientific methods that you have? Descriptive versus mechanistic science, so again looking at the 3 R's and how those interplay. And then lastly, it's in data we trust, so looking at quantitative versus qualitative data for evidence-based decision making.
And so the second course that that's part of like the core 3 that many of the students in our department take is Anatomy of Scientific Error. And it's making mistakes. It's talking about mistakes, whether it's just a mistake that you make as humans, but you have to be aware that experiments and studies that are published can have errors as well. And sometimes these errors can be the source of innovation, like the serendipity, but it can be a source of retractions of those scientific articles as well. And so for this course, we examine sources of error like misconduct, honest mistakes, or methodological / systematic errors. And then we want to look at how this applies, how we apply methodological and mathematical approaches to real world examples to analyze errors that cause problems in science across disciplines. We want to explore, review, and retraction mechanisms for journal articles, which is really useful skill as a scientist, like moving forward from this course for the rest of your career. It's also a method of science self-correction too. And then also discuss historical and contemporary cases where errors constitute a source of innovation.
And so similar to the structure of How Do We Know, each week the students would have different discussion topics related to academic ethics and sources of error in scientific practice. So starting with like a lack of rigor, which errors can be traced back to non-rigorous study design and conduct? How do we reduce or avoid errors resulting in non-rigorous science? Reforming science, what cultural and value-based structural and policy reforms can help promote research ethics and integrity? Responsible scientific communication, what elements comprise ethical scientific communication, reproducibility for everyone, what is open science, how can open science guidelines help tackle the reproducibility crisis in the scientific enterprise, and then meta science, how do you conduct the science of science, studying science, how can collaborative meta research help us develop better evidence-based practices and policies. And then the final course that we'll talk about today, which is like, the big 3 is causation. And so causation is one of the central concepts of science across all biomedical and public health disciplines. And this course is really delving into understanding the causal relationships in these different disciplines, how it differs, and then kind of understanding why things happen, predict what will happen, and then really engaging in evidence-based decision-making for like what is a true causal relationship. And so this course is designed to acquaint students with the central concept of causation across these different disciplines, discuss how cause and effect relationships govern today's research and evidence-based decision-making based on social, physical, political, and economic determinants of health, compare how fields and disciplines in biomedicine and public health approach causation differently, and use research case examples that illustrate major morbidity and mortality related health problems, which is so important for us in the School of Public Health, and then examine strategies to mitigate the limitations of causal inference. And so with this course, we actually have multiple different guest speakers that come in to discuss their research and what causation looks like for them and in their their studies. And so, like as I was saying, with the different biomedical and public health disciplines, we have like a wide range of different topics. So international health, health behavior and society, health policy and management, mental health, and then microbiology. We have these like great speakers that come in to be able to discuss what their research is, and then a paper that they select for the class, and then what is the causation in those papers look like. And so overall, upon completion of the certificate program, students will be equipped to analyze research strategies, formulate critiques of interdisciplinary literature, and apply ethical considerations in scientific practices. They'll develop skills in hypothesis testing, data visualization, error reduction, and effective communication through evidence-based strategies and storytelling. And so we really want to now shift towards like how, as students and TAs within or in like completing the R3 certificate program, what applying the 3 Rs in practice looks like. So starting with rigor, we really, you know, I think the biggest thing we like looking at our research as within like our microbiology and immunology, we have, it's very important for us to have well-defined controls, consistent methodologies, and appropriate sample sizes to enhance the validity of our results. We use very detailed step-by-step protocols to ensure that the data accurately reflects biological processes and are not skewed by technical inconsistencies. Reproducibility, with like using these detailed protocols, we also have like responsibility, as I guess in reproducibility, to share this data and make sure that the this is available to other researchers to be able to also have it and see if they can reproduce it in their hands. And then responsibility to like avoid overstating conclusions and acknowledge any limitations in our research, and then implement blinded study designs when feasible. And so like now, like these are obviously very broad for a lot, most scientific research. So now looking specifically in our lab, which again, I said we studied perinatal HIV. Kristen's going to talk about some of our like specific instances of.
Yeah, so I can kind of break it down first into like rigor and reproducibility. So in our lab, like we have a really, I would say it's a very tight ship where like our samples, at least Sumia studies samples from like neonates, so infants. So there's a lot of kind of eyes, and there's a lot of responsibility of who owns these samples. So each sample has to be accounted for. And I think, like us being in this lab, we have the opportunity to practice really strict rigor and reproducibility. But this goes for all labs, even if you don't work with, human samples and you work in like a mouse study, or you just work in cell lines. It's the same concept. But we always maintain like quality control. Like there's multiple eyes on each sample at all times to make sure that it's not one person's faults for mistakes, that we're kind of a team when it comes to that. We have like very large data sets that we have, and multiple people are doing experiments that go into this data set. So like if there's a mistake in the data set, it's not just one person, it's the team that has to figure out how to find the mistake and make sure it doesn't happen again. And so I think we have a lot of practice in our lab regards to that, and just like that the samples, they're pretty rare to get, and they're we want to make sure we use it to the best of our ability, and we always make sure that there's a lot of practice involved before using these samples. So that's where the rigor comes in, to make sure that we're passing our own QC, our own
QC.
training to be able to handle
The
certain
precious
samples.
samples.
Yeah. And so this is I just pulled this slide from some of the slide from our PI, but one of the network that our lab is a part of is called Impact. So I think this is good. Again, as I had said before in the student panel, like at least in the HIV field, there's a lot of community involved in it. And I think this really illustrates how vast, this network is. Like, this is only one of the networks that we work with, but that in each site, each clinical cure trial site, country, those are people living there, and there's communities there who don't really, they might be apprehensive to joining these trials, but yet, we need people to participate in these trials to be able to continue our research with those samples. So I think it's really important to humanize that as well, to say like, you're in the hood working with cells, these come from somebody, and they agreed to give their cells to you. And so you want to, respect that and do justice by it, and not, make sure that the cells don't die, and you get data from it, because they're very precious, and that's people's lives that they have allowed you to use. This kind of is bridging onto the responsibility of things. And this is why I kind of broke out responsibility on its own. As I was saying before in the student panel, I think we we also have the opportunity to have a huge sense of responsibility, specifically because we work in the perinatal field of HIV, where these are kids. Sumia works with 48-hour-old infants, so they're very young, and the consent process with the mothers is actually, it's very it's very laborious too, to get things in order for these clinical trials. I think it's so important for, at least for people who do like human research studies, to be able to interact with those people who then you can directly like affect, so like if you work with, like for us with HIV, we can talk to people, shake people's hands who live with HIV and see how life is in their eyes. And it's actually interesting. There was one conference that we had that had people living with perinatal HIV, and some of them were saying that their friends who also live with perinatal HIV are even scared to get a cure, or if a cure even came, they weren't even sure if they would want it, because it's something that's so
Intrinsic.
yeah, like that's part of their identity. Like they were born with this. They only know life as someone as having HIV. So they're almost scared to take that step of what would a cure look like, and how would that, it's going to change me. And there's a lot of things with religion as well, of like going, because HIV is integrated into your genome, it's like changing your actual genome, and removing HIV, like that's removing a piece of yourself, even though someone like, scientists might think that's crazy. Like, of course you would want a cure for HIV. Like, why would you be apprehensive for it? So I think this responsibility and to connect with the community of people that you are researching, if that is the type of field that you're in, is so important. And I think it is the most important aspect of this as well. They all go hand in hand, but in order for your own sense of self to keep up the rigor and keep up the reproducibility, I think it really, the cornerstone is the sense of, I have to do this right, and I'm doing this on behalf of others. And yeah, so that's I actually included one other slide too. I talked about this during the student panel, but something, at least with the HIV field, they're really trying to crack down on people using stigmatizing terms. So I included some examples here. And unfortunately, even a couple months ago during our annual meeting, there are still, I would say older PIs who still use stigmatizing language, but I think we're getting better at, calling people out to change that. And I think this all goes back into the community and the responsibility as a researcher to keep things human and respect for the science that you're doing. And with that, yeah, that is.
Lovely. Thank you so much, Kristen and Sumina. And now I'd like to introduce Dr. Billy Mills. He is at the Mount St. Mary's University. The congratulations, he's now moving on as an assistant professor, and he would like to talk about taking OUR3 into the undergraduate classroom. So
can you see and hear my slide, or see my slides and hear me? Cool.
yes,
Thanks, Maki,
thank
for the
you.
introduction, and thank you so much for the invitation to share with you guys. So as was said, my name is Billy, and I'm currently an assistant professor at the Mount, but I'll give you a little bit of background to get there. And particularly what I'm going to be talking about today is how I'm currently planning to and bringing the R3 material that I learned when I completed the certificate into my teaching. Just a little bit of a timeline to kind of put into context where and when I was completing the certificate relative to my training and my job now. So I did my PhD at the School of Medicine and Biological Chemistry from 2017 to 2023. And then right at the tail end of that is when I engaged with the certificate. But I also wanted to throw in here that I was introduced to and got my first exposure to teaching as an adjunct instructor at Stevenson University, which is just north of Baltimore, in the second half of my PhD. And so I thought the blend of being exposed to teaching and working with undergraduates while I was also engaging with the R3 material in the certificate program was kind of this perfect blend of topics that are important for research for a demographic of student that really needs to learn this at that point in time to get there. And as was said, I'm now an assistant professor at Mount St. Mary's in Northern Maryland, just in my second year here teaching. Kristen and Sumia gave a wonderful introduction of these courses and what they entail, so I don't think I have anything to add to them, just to show you the courses that I took when I completed the certificate, and I have slightly different names than they do now. But all of these topics were so useful, I wouldn't have removed any of them from the ones that I took. Things like how to write clearly for scientific writing, quantitative reasoning, scientific error, how we actually do science, reading papers, and causation. But those were also well explained. I won't add too much to them, but just to say this was my experience in the courses that I took. All right, so some of the big lessons and things that I learned when I was completing the certificate, and then shortly I'll explain how I use them practically, was one, how to design experiments. Had some exposure to this, of course, in introductory courses, but never actually doing my own experiments as I did in graduate school. So learning how to do that effectively and responsibly was useful. Some statistics and data analysis. Again, this is kind of a skill that at least I felt, dropped into when you begin graduate school, not something you get a lot of instruction on beforehand, so that was useful. How to interpret scientific literature. Similarly, maybe I'd read a couple papers when I was in undergrad, but reading scientific papers regularly in graduate school is just something you're dropped into, and getting a little bit of exposure about how to do it methodically and to glean as much information as you can from those papers was really useful in that course. And then probably most relevantly to my current role as an instructor at a primarily undergraduate institution is how to tell stories about science. One of the things that's really apparent for anybody who has done any level of teaching is that usually, maybe as a student or as a teacher, you've experienced that things stick with you more when you hear them in the context of a story. So just telling somebody the structure of DNA is not really as compelling as telling them the story of Watson and Crick and how they use Rosalind Franklin's images to figure out that structure. Or one of my favorite stories, the discovery that peptic ulcers are caused by little bacteria in your stomach. That's a fact that's maybe useful, but if you know the story that the scientist Barry Marshall drank a solution of the bacteria to give himself peptic ulcers to treat himself to prove it before he won the Nobel Prize, is I think a much better story and one that I'll never forget. So in the certificate program, learning how to and trained how to talk about science in a way that's engaging both for scientists and non-scientists was incredibly useful, and one that I'm applying all the time. And then, of course, just making science reproducible. How do you ensure that the experiments that you do as a scientist in graduate school and beyond can be redone by other researchers after you? And so a very practical example of how I was able to apply this in my research was my first author research publication when I was in grad school. And this was a tool that we developed to help process a certain type of RNA sequencing data. We were studying microRNAs, and Gautam had shared in the student session that one of his frustrations was the reproducibility of code. That's a frustration I shared as well, and many researchers do. And one of the problems I had was when I was setting out to process the data that we were generating, there were essentially no tools, references, guides, any really useful information for how to do it. Most people, if you've engaged in computation or coding, you might see in the method section of a paper, data were analyzed as described previously. And they reference you back to a paper that references another paper, and you never actually see the code that they used to run it. They might say they used a particular tool like bed tools or Bowtie in sequencing, but there's a lot of ways you can run those tools. There's a lot of intricacies to the code, and it's becoming more common, but most researchers don't include those nitty-gritty details in their publications. So that was a frustration for me as I was teaching myself to code and trying to do this on my own. So when we made this tool, we wanted it to be as reproducible as possible. We wanted you to be able to use it, know exactly how and why you were using it, and then for other researchers to analyze your data in the exact same way that you did to see if their results were similar. So we wanted to make it as reproducible as possible. And we also wanted it to be a training tool. Like I said, I had no computational training prior to graduate school. And so this was all self-taught. So if you ever use our tool or look at it, you'll see hopefully that we've annotated it so well that even if you've never coded once in your life, you can at least understand how the tool works the way that it does. And the only reason I bring this up is not to get you to know the specifics of the tool, but just to say that those principles and skills that I learned in the R3 certificate were directly applied to making a tool like this. How can I make this science as rigorous and reproducible as possible for people who might use it in the future? Like I said, during my time in graduate school was when I was first exposed to teaching undergraduate students, and it's what I do now full time. And what I observed throughout my time with undergraduates is there are certain skills that are simply lacking that not only are useful and necessary for undergraduate, but if they're going to move on to graduate school, they're absolutely essential skills that I thought they weren't quite getting the training that they need to succeed in. And some of the major ones were data visualization. I'd say the majority of students don't even know how to use Excel effectively. To no fault of their own, they were just never shown how to use that tool. As most of you who are in graduate school know, Excel is probably not a tool you're using very often. Your data sets are far too big to be using this, but I think it's an excellent way to at least introduce basic concepts of how do you visualize data? What are your variables? How do you, why would a pie chart be better than a bar graph for certain things? So that's at least how we start them off as undergraduates. So they just weren't possessing that skill. Writing scientifically is a new thing that people have to learn how to do, right? A language, you don't know how to speak a language when you first are learning it. I think similarly, the way we write and talk about science is very unique. And so students, when they're writing lab reports or any sort of summaries of scientific papers, just don't quite possess the terminology, the sentence structure, and the general way of writing scientifically. And then also talking about science in a similar vein, that how we communicate science to the public, both to our peers in the classroom, and if we ever presented, say, a conference, how do you talk about science in a way that's engaging, that others will be interested in, and want to talk with you more about, or maybe collaborate in the future. So this is my observations from having spent time with undergraduates that I thought needed some addressing. And so it's interesting, right before I received the invitation, maybe less than 48 hours before I received the invitation to share here, I was thinking about what I would do for undergraduates to bring the R3 program kind of into an undergraduate setting. And there may be a few other schools doing this, but I don't think it's as common. And so I thought, what were kind of, if I had to break it down to 6 main topics that I would want students to take small little kind of module coursework in. And these are the ones that I had come up with. History of science, I think one of the most profound things for me in graduate school and in the R3 certificate program was how me doing science fits into the history of science. We're all participants in this centuries long, a millennia long pathway of uncovering the mysteries of nature. And I think it's important to put ourselves in that context. What has been known before us? How did those people figure it out? How are we shedding new insights that will impact the people that come after us? How to design an experiment? How do you pick effective controls? How do you determine the appropriate number of replicates? What's the difference between biological and technical replicates? I think those are important as students begin their own research processes. Then processing and visualizing data, as I already mentioned, that seems to be a skill that's lacking, introducing it to them in Excel, and then eventually bringing them into more computational methods. Research ethics, usually something they may have read something about in pop culture about problems with scientific research, but as these students are beginning their own research, it's important that they know at the outset, how are they going to do this ethically? And then writing scientifically, how do you write papers, lab reports, reviews, grant proposals, a really important skill. And then talking about science, and here I mean verbally, how do you give a presentation in a classroom? How do you design your slides to mesh with the things that you're saying verbally? How would you talk about this with your families and people who maybe don't have the same scientific training that you do? So these are kind of those courses. And like I said, this is just at the outset of my planning, hopefully something that I'm able to bring into practice in the next couple of years here at the Mount. And that is all I had. I've been so grateful for all the insights that shared. So if you guys have any advice for how to implement these things or any feedback, I'd love to hear it. But thanks again for the opportunity to share.
Wonderful. Thank you so much, Billy. Thank you. And now I would like to move on to the last presentation. Dr. Ahmed Ozai. Ahmed is a postdoctoral researcher at the University of Maryland School of Medicine. And the floor is yours. Thank you, Ahmed.
Thank you so much, Dr. Kobayashi. Hope you can hear me well. I'm going to share my screen. Are you able to see my slide? Okay, perfect. Thank you. So my name is Ahmed. I'm a postdoc research fellow at the Department of Neurosurgery at the University of Maryland. I also did a graduate degree at Hopkins from the Bloomberg School of Public Health. I did my MPH there. I really wanted to pursue the R3 certificate, but maybe I was taking too many certificates. I chose to do the clinical trial certificate, but anyhow. So today I wanted to touch on retractions and the case for revising and retracting outdated evidence in science to support R3. And what can we learn from Cochrane reviews as a different paradigm of retraction? This work was led by stellar medical students from across the world. We have an international team. We do these projects on the side. Tirth is a medical student in Ukraine who was affected, but still chose to do this work, and I'm really proud of him. I have no financial disclosures, but I do have a strong personal desire to tackle research misconduct, and that is my non-financial COI. So as a prologue, some of you here may have heard of this name, Dan Ariely and Francesca Gino. And this story was in the news and took over like a storm last year. This story broke that these guys who used to tell about honesty and dishonesty and ethics and had these really cool experiments, a lot of the data was fudged. And it took a lot of us by shock. Many of us, we had read Dan Ariely's books. The Predictably Irrational was such a big deal. And to see that was so shocking. Could any of you put in the chat who this person is on the slide who's depicted here? This story has just broken right now, 2 weeks ago. And I think this is the most important stories that will shape science policy this decade. Would love to see any names in the chat. Can people recognize this figure or this person just from this? No worries at all. So this is Eliezer Maslia. This guy was in charge of the portfolio of the National Institute of Aging of the NIH for all Alzheimer's disease research. And he managed a portfolio of $1.6 billion. And that's a billion with a B. And so science has been doing a long-standing investigation, and this investigation broke 2 weeks ago, and that said of his over 800 research papers that he published before he came to NIH, there are over 100 papers strong with research misconduct. It's just an incredible document. And basically NIH, fired this person immediately and said, this top government scientist engaged in research misconduct. This is all a big deal. I think, for example, Alzheimer's research has been, we consider has been truly misled. We knew of these papers, Mark Tessier-Lavigne at Stanford, Sylvia Leswain at University of Minnesota, but this is $1.6 billion. And so this is all the prologue. And so in this regard, many of us has documented that the frequency and the cognizance of retractions and withdrawals has been increasing across science and medicine. This is some work that we have done previously, appeared in neurology, and retraction watch database has been a great tool for studying this. So we have this paradigm right now, retractions and misconduct, that research misconduct, the ultimate result is a retraction and, downstream sanctions. And the vice versa, that if you have a retraction, you basically have a misconduct. Generally, that's how we see things. And if you can see, this is Dan Ariely's retraction that has happened, and more retractions are propping up. Most of these cool psychological findings, unfortunately, were found to be fake, false, or fraud. So can there be a different paradigm? Some of you who may just be starting out in the, looking at retractions, I would encourage you to sign up for a blog called Retraction Watch. But fundamentally, can there be a different paradigm, and how can such a paradigm tackle outdated evidence, which, some of our work tackles? And so for example, for this work that we did recently is Cochrane reviews. So Cochrane reviews, for those who may not be aware, are highly influential evidence syntheses that guide policy and practice globally. And Cochrane has uniquely strong policies related to not only evidence generation, but also outdated evidence. And such a policy can potentially ensure R3 for clinical evidence. But so far, nobody had ever looked at retractions and withdrawals of Cochrane reviews, and how does that can be put into context of retractions broader and scientific integrity. So this was a retrospective meta-scientific study we carried out recently using PubMed, following STROBE guidelines. And for this work, we included Cochrane reviews published from inception to December 2023. So we systematically searched for and evaluated withdrawals and retractions of Cochrane reviews. We excluded protocols. We extracted data with independent review and validation. We looked at the year of publication, the country, what Cochrane editorial group publishes that review, the World Bank income classification of the country from which the review originated, and the listed reasons. And when I say review, I don't mean this is not a narrative review. This is an incredible body of work that a Cochrane team does under very high methodological standards. It typically takes 2 to 3 years to do and publish a Cochrane review, where it's a very cumbersome, painful process, and the final document is around 80 pages long. So it's quite a bit of work. So here in this meta-science study, what did we find? We had nearly 470 withdrawals and retracted Cochrane reviews, co-authored by nearly 1,700 co-authors. And of this, 30% had been withdrawn, and then they had their updated versions published later. And this peaked in 2007 and 2015. And if you see, this was well balanced across Cochrane editorial groups. So Cochrane has editorial groups which tackle certain subjects, and pregnancy, childbirth, pain, palliative supportive care, oral health. But mostly they came from high-income countries, followed by upper middle-income countries. And this is maybe reflective of the fact that Cochrane reviews themselves mostly come from high-income countries. You need to have a lot of resources to do this kind of work. So presumably it follows downstream that retractions and withdrawals also come from them. The highest come from the UK, Canada, Australia, the US, and Netherlands. And let's now look at the money slide. This is the key slide. What are the primary reasons for withdrawals and retractions in Cochrane? So if you look at the top, I've just magnified and put in the lower right corner, outdated evidence and articles being out of date is the most common listed reason. And the related reason, of course, is authors are unable to update the review. Now, what does that mean? So Cochrane is saying that you must update this work, this magnificent work that you have done every 5 years at least. And that is a lot of work. Most scientists are not asked to do that. When we publish a study, the journal doesn't come back to us and say, hey, by the way, that work you did, you need to update it now. It's been 5 years. But Cochrane does that. And if you don't do it, they basically pull back the study. They say, we can't rely on it. And if you were to say, no, we have definitively and conclusively proven that this is the literature, this is how it is, and I don't think doing an updated study will change this. It takes a lot of convincing to do that. So if you can, as you can see, nearly 50% of the reasons for a retraction at Cochrane is not research misconduct. It's basically updates and article updates and authors being unable to update the review or currently being updated. And if you see, failure to meet Cochrane's methodological standards is a big deal. We often hear about retractions being from misconduct. I have rarely ever seen a retraction notice where the journal said, we actually reread the methods or we relooked at the methods and it wasn't enough, but Cochrane can actually do that. What are some limitations of this, just as we look at this work, is we did not have a true comparator arm. It would require a much larger team to look at. And this was a non-matched design. If we truly wanted to do it, we'd have matched it to non-Cochrane reviews that are similar in scope and impact, such as systematic reviews and guidelines that appear in the NEJM or JAMA or the Lancet. but it's hard to argue for a match design in meta science studies. Now let's come to the key point. Why do I do this? Let me try to pull it all together. So the major chunk of withdrawal and retracted articles here in this work that we have done had reasons aligned with Cochrane's policy, articles being out of date, authors being unable to update them. And withdrawals and retractions due to errors in methodology, errors in analysis, or errors in data extraction were quite low. But when we look at science in general and medicine in general, that is not the case. Most of the withdrawals and retractions that we are seeing today are due to misconduct and major fraud. And Cochrane's policies are different from ICMJ policies. An article would not be retracted for the sole reason it was out of date as per ICMJ, but it could be retracted as per Cochrane, as we have just seen. And so we come to the philosophical question, the 30,000-foot view. What kind of evidence do we need to make healthcare decisions? And I just stepped out of a spine case. And in spine, as you may be or may not be aware, a lot of the interventions that we do, they're based on very poor evidence, actually. We have very few randomized trials. We don't have great data in spine. And spine surgeries, it's unclear, many of the surgeries, if they even benefit patients. So we come to this question of, do we want evidence to be static and outdated versus living and reliable, supporting the principles of R3? So this rise in retractions and withdrawals, this signals a shift towards accountability and transparency. But still, most journals publish papers as static records. Most don't use a living evidence approach. And so what I hope to perhaps argue to you and convey that this maybe we need to reform and change our perspective towards retractions. We can help tackle outdated evidence. We can help tackle evidence we may have done harmless errors. And so we can incorporate into policies, we incorporate into cultures in our labs and our groups that we need to work to ensure continued reliability in science and clinical evidence. It's not like if we publish a paper, we are done and it's done and dusted. And perhaps there should be a focus at least on regularly updating evidence. And this is a novel approach that people haven't advocated for outside of Cochrane. So just in conclusion, these are the take-home points. That in Cochrane, outdated articles and author unavailability to update evidence is common reasons for doing a retraction. And this is quite different from retractions from other journals. And this is a novel paradigm. And that retractions as a part of living evidence synthesis go beyond the traditional sense of retractions and can help ensure continued reliability of science. So thank you so much. And this is a picture of my cat.
Wow, thank you so much.
Thank you, and happy to answer any questions.
Yeah, so I just want to say a brief thank you to all of our presenters, from Matt, who is a master's student, who talked so wonderfully about his current experience and lab applications, to the dual presentation from Sumya and Kristen, who are more experienced PhD researchers at this stage now, and then Billy. It's so wonderful to see how you're, thinking about implementing this further in the undergraduate setting and expanding that work. And to Ahmad, who is a, trained physician, and seeing this application and relevance for retractions in that realm. It's been so wonderful. I do want to open it up to questions to everyone from the audience, to these speakers. And while everyone thinks of questions, I will request if everyone can turn on their cameras just for 1 minute. We do want to take kind of a screenshot of all the attendees who are currently here. That would be great. And then, yeah, we'll just move on to questions. And if you can't turn on your camera, that is okay, but maybe just for a few seconds while we capture a screenshot.
Okay, so I'm going to take the picture of us now.
And in the meantime, I wanted to thank everybody for their wonderful talks. This was absolutely outstanding. So I'm still totally flashed with everything that you have accomplished. And of course, it is wonderful to see this at multiple different educational career stages. So that's very gratifying for me too. So some of you are applying this directly to your research in the lab. Some people take it out to their PhD work, and some people already took it to professional practice. So that's something that I'm still cannot believe my luck that I have the privilege to work with all of you. So I wanted to just thank everybody also because we forgot to mention this earlier, everybody who spoke today, of course, was part of our group of awardees actually, and we are going to get back with you, of course, on this. So congratulations also on your award for the Catherine Welsh Scholarship. So something that we are very grateful for. Where are there maybe any questions from the audience? Of course, I always have questions, but I don't want to be the one who monopolizes the discussion here. So what are questions from the audience, please? Well, I give you a little bit of time to think. Going to start with Matt. Matt, you had something, you spoke wonderfully about your work, and I'm not surprised because I saw you speaking about your work a lot in the coursework that we had together. But particularly, I wanted to ask you about, how was your thinking when your PI said, well, first you look at your data yourself and you don't look at the literature? Because normally, actually, people say the opposite, right? So I found this quite eye-opening. So how did you feel getting lost in everything, or how did you approach this?
Yeah, I mean, it was definitely an interesting experience, to be honest, because that's not how I had, that's not how I had approached science before. Um, but like, I actually like sat down and was like, why did you want me to do it this way? Like, why is this? This is not nearly how science is done. And he kind of like sat down with me and like explained about like I talked about the biases, and like he wanted me to like the data that I made was valid, and he wanted me to be able to sit down and analyze my data that I had made myself and not automatically assume that it was that it was not good or valuable data. And um he also didn't want me to automatically assume that it was going to be good data either, or go into it thinking I was going to get a certain result. He wanted me to get my results first, think about it and analyze it, and then look at the data and stuff like that. And like the critical thinking aspects of being able to do your own data analysis, I think was really what he was trying to focus on more. And also like what I'm doing is it's slightly novel in the sense that like we don't a lot of people don't really take colonies that have grown on an antibiotic plate because they automatically assume that because they grow on the antibiotic plate they're resistant, right? And then go back and test their actual resistance profiles to antibiotics via MIC methods. So that's a relatively novel thing that I haven't really seen much of in literature. So I think that's why he did wanted me to do it first and think about it and then look at papers, because a lot of the MIC or like resistance profiles or like stuff like that, don't follow that approach. So I think it was more because it was a relatively novel stratagem.
Excellent. And you obviously mastered it very well. So excellent. Very
I
good.
wish. I still have trouble with it from time to time.
That's normal. That's part of science. Any other questions for Matt as we are? So I wanted to follow up with the next speakers, namely Kristen and Sumia, actually, who are not because of their personal age, but because of their exposure to the program, are veterans actually. The programs have like been with us for a long time, and in very different aspects. And particularly wanted to hone in on your very impressive reflection on the notions of responsibility and how to live it, right? And you spoke in a very moving way about your responsibility towards the patient samples, and also what I found really impressive, I might say, about the community decision to look into language, right? Especially since we spoke a lot about harassment and its impact on culture in research, right? And this is something that I found a quite impressive way of how the community has reacted to a need, obviously, right? And have you observed how this came about? Was this a community initiative, or was this something that the NIH had initiated?
I believe it was a community initiative. I don't know exactly when it really started, but I mean, in my 4 short years, or I guess almost not even 4 really, of being in the HIV field, I've seen the even just like in this short time, the progression of like how the community has really influenced the way that we communicate our research and how the language that we use surrounding our research, and not just like when we're presenting it, but when we're just like, not just in like bigger presentations, but when we're like talking every day in lab, I feel like we can really see the way that has like changed the way we think about our like what we're actually doing. I feel like I don't know exactly how, but or like when this really started, but like one of the things I thought was very cool about like the HIV field in particular was the fact that when we go to conferences, the community members are invited. They're there and they will speak up if they hear somebody using stigmatizing language. And I mean, it can be very embarrassing in the moment, but it does help really change the culture towards centering our research around that the people who are actually living with HIV. And so, while I don't know exactly if that's like where like the real influence came from, where people being invited to come to these conferences and speak on their experiences and then kind of weighing in their thoughts, if that's like where it started, or if it's more of just like the history surrounding the research and kind of how the research has like, I guess, the stigma that was so ingrained in the HIV field from like the start of that, like the epidemic. I feel like it's really kind of like the researchers trying to like right that wrong of like what we saw with like in the 80s and how stigmatizing that has still like that time has still like impacted us now. Yeah.
Yeah. Any other questions for? I think Kristen had to sign off. Is this correct? But
Yeah, she had to run, so it's just me. So Samia,
Yes,
I'm
yeah.
under Kristen Kelly's Zoom, but we share an office, so we sit right next to each other. So we just both logged on. It's just her computer, so it's just easier.
Any other question for those particular speakers? If not at the moment, then of course, Billy Mills is one of our rock stars, has always been in the program, right? But of course, we are so proud of you, of what you have achieved by moving on to, luckily still in the neighborhood. So we are glad you're still in Maryland. So still, this is in Emmetsburg, right? Where you are, right? So at least you're in driving distance. So that's something that we enjoy, of course.
Yeah.
Definitely. And of course, your idea to make the R3 certificate an adaptation for the undergrads is just brilliant. So this is wonderful. So how did you have an impression of what kind of, apart from what you talked about with storytelling and everything, but content-wise, what do you think would you need to make adaptations so that the certificate is also tangible and useful for your students? Of course, in the second year, you have a certain exposure to your learners, and from Stevenson also had a feeling for this learner group.
Mm-hmm. Yeah, it's a good question. So one of the examples I brought up, I think, highlights that is like showing the students how to use Excel, maybe applicable for undergraduates, but not for graduates. If the graduate R3, coding more applicable, larger data sets more applicable, but at least that foundation. But a lot of it, I think, would be very similar. Like storytelling, I don't think it's different between undergraduates and graduates, but they definitely need a lot more training in scientific writing. Like when we send them their first lab reports and have them write back about their first experiment, it's very clear that they've read very little and written very little about science. So we have to spend, I think, more time on that kind of structure than you would for graduate students. One idea that I and some colleagues have had is like building up to a lab report. So the first time they write a lab report, it's not a full lab report. We fill in most of the text and then they fill in the relevant pieces, and then week by week remove the amount of material that we give them, so that by the end of the semester, they're writing full lab reports. But they don't like it writing full lab reports for the first time, and we don't like grading them for the first time. So I think a better way would be kind of, we call it building up to better lab reports. So maybe those are a couple of examples of how that would be adapted for undergraduates. I wouldn't take a graduate student and teach them how to write a lab report. They're probably a little bit more advanced there.
It is something that I would say is a skill set that is underestimated on all levels, right? So we always place this in the undergrad realm, but of course, all of us went to graduate studies and know exactly how critical a skill this is in all form of communication and handing over projects. And the mentioned earlier reproducibility for everyone community actually focuses particularly on this particular issue as a major source of error in science, namely the fact that lab reports tend to be an underestimated skill set. So I definitely congratulate you on your patience and persistence in this area.
Thank you.
Any other questions for Dr. Mills? Well, if not at this point, then I would like to draw your attention to Jade's question in the chat box for Ahmad. Actually, Dr. Ozair, would you like to directly jump in and answer the question of Jade in the chat box?
Sure, thank you. And thank you so much, such a wonderful symposium. So I agree, the current publishing system is not really conducive to revising and updating, but I think things are changing. So if you have something where you want to update a paper, it's a published paper, you would ask yourself, are the findings now really wrong? Did we really do an error? Are the findings unreliable? Or maybe there is no qualitative difference and the effect estimate, the effect size is slightly different than what we had estimated. So I think you have a couple of avenues. If you have the latter, you could, simply right now you have so many web-based resources. As you pointed out, bioRxiv, medRxiv, or other options, OSF, Zenodo, or other options where you can list that, and you can link to your older paper and say, hey, we reran the analysis and this. So it doesn't really change the conclusions, but the effect size is different. Versus, something where you made a major error, and in that case, I would encourage you, and let's all accept this culture, that it's okay to make errors, but it's important to reach out preemptively and say, hey, we made an error in the analysis, and this is not reproducible, and we would like to retract it. And I think even if you have a retraction on your CV, but you can put an asterisk on your CV and say retracted voluntarily due to self-discovered error, I think nobody in science will look at it and think badly of you, for sure, at this point. But I think we are still all so afraid of getting a retraction that we think it's taboo. So, but things are changing, and I've put 2 links. There are some journals now that are become more open. F1000 Research is very forward-looking journal, and they have become quite open to revising outdated papers.
Thank you so much. And Jay, do you have a follow-up question on this, or is your question answered?
Yeah, maybe I have a comment. I believe that's more than I believe that we have to add one more R in our 3R, right? One of them is review, because when we are now it's so difficult to find good reviews for papers because the people are not willing to review anymore because we're not being paid or stuff. But I believe that review is an important part of our job. And sometimes I saw reviewers not willing to do so. So they do a bad review. And in fact, if you do some, let's say not a mistake that you are not willing to do, and the review maybe can see, but if the review do a bad job, you have bad science in the internet. So it's just one point that we all, the future PhD students, the future PhD and us, we have to do a very, very good reviews. And sometimes we have to be willing for what I tell to my students when how to read a scientific paper, and I talk about the willing to do a review. You have to study, you have to check the reference, and sometimes the reference is completely wrong, right? So we have to check in order to improve the quality of paper. So I believe that's rigor, reproducibility, responsibility, and good reviews. And a review will be another R. Just one point I'd like to add. Yeah, and I am also afraid of some stuff that happened in the US, because some institutes, you have to pay your salary. And right now they are not giving so many R01s. So I believe that's one lab had in the past 5 R01s. Now they must have they have 2, and the salaries goes down. And I'm a little concerned about how the people because some ethic problems happens when you lost your house or something like that. So I'm a little concerned about that, and I like to just expose my point of view. I'm concerned about it. You have to pay your salaries by grants. In the future, could be a problem for the science. Just a point of view. Thank you.
Yeah, Ahmad, do you wanna react?
I completely agree, and this is such a great point that you have brought up, Dr. Martins. So there is currently no incentive in a promotion and tenure committees to reward people for doing good reviews. There's absolutely, I myself am flooded with reviews. So far in my short career, I've done over 300 manuscript reviews, but then I said, this is madness. I cannot keep doing them. I have, if I don't write my own papers, my career I will never get the faculty position. I'll always remain a postdoc. So it's a huge challenge. You mentioned it. And without explicit incentive, the university saying, provided you do 10 or 20 good reviews, that will be counted as 1 point on your CV, similar to one paper, so one reasonable research paper, non-cell nature science, of course, sure. But I think we just have a mismatched incentive structure. There is an incentive to publish fancy data, and there's no disincentive to do bad research practices. People can do bad research practices, apply for these grants, as you very rightly pointed out, and unfortunately it will all go through. And now but what I'm hearing through this is something also I want to share this panel. My mentors who sit on study sections are saying the next 10 years of NIH grant funding are increasingly uncertain because the taxpayers are seeing this kind of fraud happening. And I think there is increasing disapproval of, where is the taxpayer money going? This is taxpayer money that we all use. And I think we need to hold it to a higher standard.
I would like to, there's so many people who have obviously reactions to that. Should we go in sequence of appearance, right? So Matt, I see you have your hand up, but Billy actually put his point first in the email and Ilinka just reacted. Should we just quickly jump onto that point? Billy, do you want to quickly comment?
Yeah, just briefly about the point that Antonio made about reviews, that it's amazing there's papers that don't even bother to take out the prompt that they put into GPT or the response that GPT says like about their question. Like it doesn't read like a human wrote it, and the review process is so poor that they can't even catch that. So huge problem.
Yeah. And Ilinka just put an example from Retraction Watch correspondingly in there. Yeah, I have no idea. It's incredible what people always think that they can do, right? So like it's quite interesting. Yeah, thank you for that. Marie had a reaction also. Marie, are you still here? No, I'm not sure. She is probably not here, but she mentioned that some published classics that only finally be appreciated after many years, right? So yeah, that's an issue, right? Indeed, of the scientific landscape, right? Indeed. Matt, you also wanted to comment. You have your hands up.
Yeah, so I feel like especially with like the chat GPT and like the problem of a lot more like lack of review on the like journal portion of the stuff, a lot of the onus for figuring out what is good research and what isn't good research falls on the readers more so nowadays. And like and that also I think kind of ties back into what I was talking about, how like you do your research, you find your results, and then you can look at literature, and then I think you can kind of think critically about those kinds of things. Because like, if you base your work off of like a crappy or like a fake article that didn't properly go through peer review, and like you're like, oh, my results are totally different. This isn't what it's saying. Am I wrong? Like you have to like kind of think about those things as well, like, and keep an open mind. Like that's why I was saying it's not the end all be all. It's not perfectly set in stone.
Yeah, maybe we should building on what you said, Antonio, about the fourth R. Some of us say that there should be 5 R's, right? But I mean, resilience should be another R, right? We all definitely, I think we need this when making good science, when we see how sometimes science can go astray and is not really done well. But then we all get reminded that the R3 or R5, whatever community is a community, right? So it's a community of us feeling devoted to those values and norms. And this is, what symposia like this, and basically this community that you're all part of and that you're all forming gives me so much hope, because all of what we are all doing, we carry this forward to our mentees, right? To the people that we will work with, and they will carry it forward too. I always say, like, even if only 10% of those people that whom we work with carry this forward, there's quite a bit of a catalytic factor here, right? So, like, I think we should definitely be positive about this. And basically, the day-to-day, provided me with a lot of joy, but also with a lot of hope for the future. So I would like to thank everybody for your outstanding work. Please keep on carrying the message forward. You're doing such great work, such a wonderful service to the scientific community, and most importantly, to humanity. So you're all doing important things every day, and I would like you to be aware of that each time when you are sitting down and are exhausted maybe by what you have to do and everything you have accomplished, but try to appreciate yourself for what you're doing. So this is something that's very important and outstanding work and service. Yeah, so I hope that you all can at least for some minutes join us tomorrow also, because Tamaki has already mentioned that tomorrow our day is devoted to communication and combating misinformation. And yeah, so I hope you have time to zoom in a little bit. I wanted to also mention that, of course, we are recording this. We will need a couple of weeks, maybe even 1 or 2 months, in order to get all the permissions from our attendees to use the material, if you want to give us those, and then also cut the long video into smaller parts. But as soon as this is the case, these videos that have received permission from the speakers to being used will appear on our YouTube channel, and then also on our website. So basically, if you want to use any of this material in your work, of course you can, right? So we will probably ask for a Creative Commons license so that it can be used for your educational purposes as well. Okay, well, I want to thank everybody for this wonderful first day of our symposium. Thank you very much for coming and for contributing. You have all been outstanding. Thank you so much.
Thank you.
Thank you so much.
Tomorrow. So tomorrow.
Thank you, everyone. See you.
Bye.